I'm going to talk about a recent ongoing project in my research lab focusing on SARS-CoV-2 ultra-sensitive antigen detection. So as we have heard in previous talks, there are multiple different strategies where we can use to test for SARS-CoV-2. The first large category is virology testing. So this aims for testing for virus itself, and that includes the RNA molecule, which we typically call molecular approaches. And there's a second virology testing that aims for protein detection of the virus, which is what we call antigen detection. And we have also heard about talks on serology testing, and this focuses on antibody responses to virus and detecting of the antibodies generated by the human body. So according to CDC data, as of July 5th, there are about 36 million molecular tests that have been performed and results reported, and the positivity rate is about 9%. And this corresponds very nicely with what we see in clinical testing. So here in the Pepper Clinical Lab, we started to offer clinical serology testing about 2 months ago. And we have about over 5,000 clinical tests being performed, and the cumulative positivity rate so far is about 5%. So again, this is as of July 6th, which is very similar to the overall molecular positivity rate in Pennsylvania. So these also reflect what Scott Hensley earlier talked about in his prevalence work as well. So when it comes to diagnostic testing for SARS-CoV-2, one important thing that I think we need to remember is that the test sensitivity, specificity, and the predictive values are very important to consider when we try to interpret the results in different patient populations. In a very symptomatic population, which is typically what we are aiming to gear towards detecting in the pandemic right now, sensitivity is very important in minimizing any false negatives to ensure that we catch every single positive case and to ensure that we can do contact tracing, proper isolation as well. So when we talk about sensitivity and specificity, one of the things to point out is that there are many reports that have pointed out the existence of false negatives by various methods. So depending on the literature we look at, and I listed several in the boxes here, by the end of the slide, you can see that many different reports report that up to 50% false negatives can be detected if you compare a molecular result with the clinical diagnosis based on contact history of the patient and the chest CT. And another Annals in Internal Medicine paper have looked at a meta-analysis of all the testing results so far, and they pointed out that the false negative rates may be between 100% before symptom onset to 20% minimal at the peak of symptom presentation to rising again after the peak of the presentation. So depending on the timing in disease course, the false negative rate would vary. There are other studies from, for example, from NYU that points out that rapid molecular test may miss up to 48% or half of positives detected by another molecular platform. There are other FDA EUA authorized, for example, there's one test that has been FDA EUA approved, Antigen Assay. As of this morning, there's a second one also approved by FDA EUA for antigen detection that uses traditional lateral flow technology for detecting the viral antigens that exhibit about only 80% sensitivity, which is limited by really the technology itself, because lateral flow is known to generate a limited analytical sensitivity. So what we can see here in what we have discussed so far is there is a need for more testing. Of course, one aspect is testing capacity. We need more testing capacity to make sure that everybody needs to be tested, can be tested. At the same time, we also need to focus on the quality of the testing to make sure that we are expanding the capacity with accurate, safe, and convenient methods. So there are several unmet needs here. We have a limited supply chain for many of the molecular methods, the collection, swabs, buffers, as well as many probes and primers are limited because essentially globally, every single country and many labs are using these supplies. And serology tests do not detect acute infection in many cases, and current portable methods generate high false negatives. So based on these unmet needs, we sort of pivot a little bit and focus on a technology we developed in the lab and try to develop point-of-care antigen detection methods to complement current existing diagnostic methods. And the key, as we mentioned here, is high clinical sensitivity to make sure every single case can be identified. So shown on the slide here is a diagram, is a picture depicting what the ideal eventual device would look like. As we can see here, we would take the typical sample, in this case, a nasopharyngeal swab sample, and have a cartridge here that integrates all the needed reagents and washing and incubation steps within the cartridge. And then once the reaction is completed within the cartridge, then in the middle of the device, there would be a window that displays the readout device, which is a microwell array that displays microbubble images. We can then capture the microbubble images and then process them using a computer vision-based algorithm and give you a test result of either positive or negative. So the feature here that we want to achieve is ultra sensitivity. They're portable, fast, compatible with a smartphone readout, and also compatible with self-testing potentially by the patients at home. So this technology relies on something we developed in my research lab called microbubbling digital assay. Hui Chen, who is a postdoc in my lab, was really key to development of the technology, and this was published in 2019. So as you can see in the core, in the center of the slide here, this is really a special ELISA assay where we have primary antibodies conjugated to magnetic nanoparticles, which will then bind to the target in the samples. In this case, would be a viral antigen. And then a secondary antibody would come in and form a sandwich complex, which then are conjugated to a platinum nanoparticle. We then transfer the entire complex to a microwell chip. On the chip, we have many microwells that contains a very small amount of liquids. And therefore, the sandwich complexes are then partitioned into these very small microwells. When hydrogen peroxide solution is added, the platinum nanoparticle then catalyzes the degradation to generate oxygen. And oxygen then get concentrated in these microwells and eventually form microbubbles, which is what we want as the readout signal. So again, the feature here is that because of the local concentration of analyte and sandwich complexes, we have very, very high sensitivity. The signaling can be very fast. The microbubble generation is within minutes. And the assay is also temperature tolerant and lighting tolerant, as I will show you in the next few slides. So we went ahead to characterize the robustness of the assay, making sure that, again, our target is to develop a robust and accurate assay. So on the lower, on the upper left corner here, you can see this is the kinetics of the microbubble generation after we incubate in the microwell chip for several minutes. So from 1.5 minutes all the way to 9 minutes, you can see the microbubble generated gradually. And on the right side, you can see these are the traces of individual microbubbles. And once generated, they remain stable for up to half an hour, allowing a very long time for imaging and analysis. On the lower right corner here, these are experiments where we try to characterize whether the microbubble generation or growth process is temperature independent. Because again, if we want to use this in either a lab environment or especially important in a point of care environment, then the resistance to different temperature impact is very important. So as you can see on the left in panel A here, the rate at different temperature from 4 degrees, 22 all the way to 32 degrees is quite constant for microbubble generation. And this is also quantified as bar graph on the right as well. We then generate a computer vision-based algorithm to quantify or read out the assay result. Again, if we want to use this by an end user who may not be a lab technologist or a researcher, then we need to have a user-friendly way for them to read out the results. So to do this, we collaborated with the GRASP lab here at Penn and come up with a two-level CNN algorithm to identify and quantify the results. So as shown here, the first level comes in to crop out the image area with the microbubbles, and the second level comes in to count the number of microbubbles. And the lower left corner here shows you the interface of the algorithm we use for the counting. And panel C here shows the correlation between the algorithm counting and the manual counting, which shows a pretty good correlation. On the right side, you can see that we also characterize the performance of the algorithm under different illumination conditions. Because again, if the device is placed under a point of care context, then there could be different illumination existing in a particular environment. So we can see that under all these illumination conditions, we can still get robust performance and accurate counting of the microbubbles. So then we proceed to characterize the sensitivity for the assay. To do this, this was a work before the pandemic where we leveraged the technology to quantify the level of PSA. So the idea is to measure the PSA concentration in the blood samples from patients who are diagnosed with prostate cancer and a post-prostatectomy. Any very early rise in PSA concentration would indicate that the patient may possibly have a recurrence of the cancer. So we, on the left side, you can see we used a different PSA concentration and counted the number of microbubbles and come up with an LOD of about 0.06 picograms per mil. We then compared the PSA concentration generated by our assay against a clinical lab gold standard assay by testing several different patient samples. As you can see, the results correlated very well between the two assays. However, our assay has about 200-fold higher sensitivity than the gold standard. So for SARS-CoV-2, we leveraged the same concept here by targeting the N antigen, which is nucleic capsid, and having two antibodies binding to N antigen. So you can see the bubble image is generated at different concentration of N, and in the middle panel here, this is a screening of different antibody pairs we conducted to identify the pair with the highest sensitivity. And on the right side, you can see with the leading pair we identified, the LOD is about less than 1 picograms per mil for that antigen. So then we went ahead and tested some inactivated cultured virus. So this is again provided by Sarah Cherry, who generously provided her cultured virus from BSL-3 and inactivated the virus and then gave to us. So we can, as you can see on the left side, as the concentration of virus increases, we can see the increase in the microbubbles. The limit of detection is about 85 copies per mil, which if you compare to the table on the right, you can see that it's at least as good or even better than many of the current molecular assays using RT-PCR on the market. We then went ahead and started to test some clinical swab samples. So on the left side, you can see these are two images with SARS-CoV-2 positive samples which have been detected by RT-PCR with either detergent or no detergent present. And in both cases, you can see there is strong microbubble signals. Right side is one on the higher upper right side. This is one negative sample that is tested by our assay, shows very clean background. And the lower right side is the cultured SARS-CoV-2 virus. So the next step for us is to continue with the clinical validation. One special focus we are working on right now as of today is to improve the specificity, because we've seen increased mucus from many of the nasopharyngeal swab samples, and that caused increased background in our assay. So we are working hard to try to improve the specificity for the assay. We're also exploring other sample types, for example, saliva, which may be able to give us a different matrix background. We're also working with industry partners for automation and miniaturization of the assay. And eventually, we want to use the assay also as a tool to study the distribution dynamics and associated disease implications for SARS-CoV-2 antigens. So with that, I'd like to thank my lab members to work very hard on this project, and my collaborators Susan and Sari have provided me with inactivated virus samples. Laurel Glazer has provided me with some clinical samples, and my dental school collaborators, Michelle and Pat, in collaborating and providing saliva samples. Our work is funded by PCPM, Penn Health Tech, and Penn Dental Medicine at the stage.